All right, well, welcome everybody to the Gladstone Newton series. My name is Deepak Srivastava, and I'm the president of the Gladstone Institutes. And we've started this Newton series that's inspired by Isaac Newton's remarkable intellectual output during the quarantine of the Great London Plague, which was in 1665. And we thought that all of you are hearing a lot about COVID-19 in the news, and we're doing a lot of programming on COVID-19 with our work at Gladstone in that area. But we wanted to make sure that you are also getting to hear about all the other areas of science that we're working on at Gladstone as part of our Gladstone family. And so we've done 4 of these now in the last several weeks on different topical areas that we're studying at Gladstone, each featuring some of the big ideas that our Gladstone scientists are pursuing. Now, I think many of you on the webinar are familiar with Gladstone, but it's either that there are a few folks that are new to us. So just briefly, for those of you who are new to us, the Gladstone Institutes is a biomedical research institute that's nonprofit that's located in the Mission Bay campus in San Francisco and is very closely affiliated with UC San Francisco Medical Center, but is in fact fiscally and administratively independent. So we have a very close scientific relationship with UCSF, which is a huge strength. But the independent nature of us with about 500 employees makes it so that we can create a structure that is often quite nimble and entrepreneurial in how we go about our science. And our science is really focused around a few thematic areas where we build teams of scientists to tackle some of the major unsolved problems around human disease. And we study many diseases, but have significant focuses around neurodegenerative diseases, cardiovascular diseases, viral and immunologic disorders, and we weave together across those a deep use of computational approaches because science is more and more driven now by the ability to acquire large data around the question asked. But the biggest challenge now is actually not in acquisition of data, but rather the creative ways one might leverage those data to learn and answer complicated questions. And so with that in mind, 2 years ago, we started an institute around data science and biotechnology that's led by Katie Pollard, our speaker here today. And so Katie is going to speak today about the microbiome, which is one of the areas where she's applied very interesting computational analyses to analyze the genomic data around our microbiome. And she's been a leader in this area internationally. And so she's going to give about 15 to 20 minutes of comments, after which we'll have ample time for question and answers. And I'd encourage you to use the chat function on your Zoom screen to submit questions, and I'll moderate a question and answer period with Katie after her comments. Now, just a brief introduction of Katie. She is, as I mentioned, the director of our institute around data sciences and biotechnology. And she's also been an investigator at Gladstone since 2008. And one of my most important moves since I've been at Gladstone since 2005 was in fact to recruit Katie to Gladstone. And she's made us a leader in this area over the years. She's also a professor in epidemiology and biostatistics at UCSF and is director of the bioinformatics graduate program at UCSF. She also is an investigator at the Chan Zuckerberg Biohub. And by way of background, she received her undergraduate degree at Pomona College, received her PhD degree in biostatistics at Berkeley, and has really emerged as a leader internationally in her field. And so with that, let me turn it over to Katie, who will tell you about her really interesting work around the microbiome. Katie?
Hi, everybody. Thanks for joining. me today during the lunch hour. It's a pleasure to have this time with you online. Going to start my talk with this beautiful picture of poop. So this is a very close-up image of a poop sample, and it was taken with an electron micrograph, which lets us look at individual cells, and the cells have been colored so that you can see that there are many different types of microbes here. And in fact, what dominates the picture are the microbial cells and not the poop per se. So what's there in that picture? What we've learned is that our bodies, not just stool samples, but our whole GI tract, our respiratory tract, and indeed many other sites on the human body are full of bacteria, archaea, fungi, viruses, and other types of microbes. They make up about 5 pounds of your body weight, and the majority of that is in the gut, meaning that there's more cells in the gut than other parts of the body, even though they're found in many parts. And what I find really astonishing is that if you think of all the proteins in your body right now, there are about 300 times more that are coming from the cells of microbes compared to the human cells in your body. And these proteins and the microbes that encode them are not just along for the ride. They're using the proteins to communicate and exchange information with human cells. They're integral to our immune system. They transform things like diet and drugs that we put into our body in ways that our human cells can't. So they produce energy and molecules and vitamins, and in some cases toxic compounds from what we put in our body that wouldn't happen if they weren't there. And therefore, I think about it as sort of an extension of our human genome. It's another part of the DNA component of our body. And it really needs to be considered in concert with human DNA, which my lab also studies to understand who we are and what makes us sick. And indeed, what makes us sick has been a big focus of microbiome research. Out of the approximately 40,000 scientific peer-reviewed articles that have been written about human-associated microbes over the last 20 years, since this field has come to light, over half of them are reporting an association between our microbes and a number of different diseases shown here. This is fascinating to me as a scientist. It's also very important to me and to my family because several members of my family suffer from immune conditions. This is my husband and my kids with whom I've been spending a lot of time lately, like many of you may have been with your household members. And I'm just noting here that I have 2 fairly serious chronic inflammatory diseases, as well as I suffered from allergy and asthma as a kid. Unfortunately, a couple years ago, my son Ellis was diagnosed with very severe pediatric Crohn's disease, a related condition. But interestingly, even though we're all living together and I can guarantee exchanging a lot of microbes, my husband and my son Ellis's twin brother, Jasper, do not have any inflammatory disease. So to get to the bottom of this, being in San Francisco, we've used all the latest, most modern techniques for precision medicine, looking at our genomes, trying to understand not just why we're sick, but also how to specifically treat us, to find a treatment that would be precise and useful for me and for my son. And I can say, as a mom, as a patient, and also as a scientist, this is not enough. It doesn't work in a lot of cases, including in my son Ellis's case. A lot of the treatments that would be recommended based on looking at the human cells in his body have not worked at all for him. And so the vision that I want to share with you today is that perhaps we could be doing better as scientists, as clinicians, and do better for ourselves as patients if this equation for precision medicine also included careful, precise measurements of the microbiome. If that were true, we might be able to better diagnose or choose the right therapies, and we may even be able to use this information as part of the therapy itself. So to track bad microbes, to make sure the good microbes are there, and to perhaps even make microbiome-based drugs or other therapies. So how do we study the microbiome? There are two main ways. The traditional one is to isolate and grow species one at a time in the lab. If you get a petri dish and it contains precisely one type of microbe, and if you can grow it in the lab, which is often not the case, you can learn a lot about it. But this is low throughput, and there are many microbes for which this approach fails. The technique that came on the scene around 2006 and has really made this field of microbiome research explode is that we could study all the species in parallel from a sample, like the stool sample, using genomic technologies. So what does that mean? We take a sample. It could be saliva, it could be lung aspirate, it could be stool, it could be a skin swab, a nasal swab. We extract the DNA from it. You can also extract RNA or proteins or small molecules from it as well. I'm going to focus on DNA today, but we're working on all four of those areas. And then you sequence it. And what comes out of the sequencer are a whole bunch of strings of letters of A's, C's, T's, and G's, and then you need to interpret those. And that's where my lab comes in with a really hard problem. So this interpretation of this pool of sequences is a bigger and much harder problem than understanding sequencing of human cells. And let me try to explain why. First of all, this test tube doesn't just contain DNA from one species. It's a mixture of hundreds, sometimes even thousands of different species. And for each one of these, say 50 million sequences of about 300 letters or nucleotides, A's, C's, T's, and G's, we don't know which species it came from. We don't know which part of the genome it came from. And so how do we make sense of this? Luckily, we do have the data from studying individual microbes where we do know the source. We know where the DNA came from because we took it from a pure culture of a single microbe. And amazingly, almost 300,000 different species have been studied in this way, one at a time to date. And each of them for a microbial genome has a sequence of A's, C's, T's, and G's that's about 5 million letters long. So we've got 50 million of these 300 letter sequences, and we want to compare per sample, and we might look at a lot of samples, and then we want to compare each one of those to this reference data or knowledge base that has about 300,000 5 million letter sequences. So this is something you can write computer code to do. It's a pattern matching problem. But it's actually a very hard one and very computationally intensive to do. What do I mean by that? Why is it hard? We had to solve a couple of really major problems to even make this feasible. The first one is that when we started doing this work, less than half of the species that you'd want to analyze had a genome sequence. So we'd have lots of data on it, but we'd have no idea what it was. And I'm very proud that a really concerted effort from my group and many others has gotten that down below 10%. And I expect it's going to, for the samples like human stool that have been studied very well so far, I think that we're going to get that down close to zero. A bigger hurdle, however, besides having the right data to interpret these sequencing experiments, has been how hard it is to do this huge pattern matching problem. And I don't know if I'm proud or embarrassed to say that this was a big enough and expensive enough problem. It got Mark Zuckerberg's attention last October when the cloud computing bill from my lab hit his desk, and he was amazed that we were spending over $10,000 a month on performing this task that I just described. And he actually called a press conference in order to complain about how expensive this computing is. And whether it's done on AWS or some other part of the cloud, or if we do it on our own computers at Gladstone, the fact of the matter is it's a lot of calculations and it is expensive to do. What I'm excited to report is that just recently, and we'll be publishing this soon, my lab has figured out some algorithms to speed up this problem. And instead of using tens of thousands of dollars of cloud computing, this task can now run on a laptop. And this is going to be a huge win, not just for us and my bill, but it really makes this technique accessible to labs that wouldn't have been able to do it previously. So around the world, especially in countries where there's a lot of disease, including infectious disease. This would be very empowering to be able to conduct these analyses on an affordable computer and in-house. And finally, there's the problem that if we have good reference data, we get the calculations done, there's still a lot of noise and complexity in these data sets. And so if you want to, at the end, say that a particular sequence is telling us something about a disease, how can you come to that conclusion? How can you say that with certainty, given the scale and complexity of this data? And this is where statistical models come in, which is another major area in my lab. So what kind of models do we use on the microbiome? One of the workhorses of microbiome research is actually modeling techniques from ecology that have been used for years and years to describe the species that are present in environments and how much of each one. And we literally basically treat each person like a different forest, and you are going to ask the same questions that a natural ecologist would ask about whether you find a particular bird species. How often do you see it? Is it more often in certain kinds of forests than others? And so these techniques have been very powerful. But unfortunately, just saying that the forest has a bird in it isn't really enough. Even to say a person has a microbe in them isn't enough to link it to disease. Let me give you two examples that I know we're all familiar with. One is that different strains of the same species can be a pathogen or not. Almost all of us have some E. coli in us right now, and we're feeling fine. But we are aware that you hear from time to time about a food poisoning outbreak with E. coli on some bad spinach or something like that. And these are the same species as the E. coli that are in all of us right now. All that's happened is a small genetic change in their genome that flips them into a pathogen that makes you very sick. We're also all aware of antibiotic resistance and what a big problem that is today. And the fact that cells of the same species, say Staph aureus, you can have some that respond right away to antibiotics, and others are multiple resistant, meaning a variety of different drugs they're resistant to. And that, again, is addition or subtraction of pieces of DNA from their genomes. So we also use, therefore, the techniques from evolution, not just from ecology, but from evolutionary biology to study the microbiome. And these are, I think, the more powerful models for trying to get at precision microbiome medicine. And the reason I think they're more powerful is that every day the cells in our microbiome are dividing. They're dying and they're reproducing. And every time that happens, mutations get inserted. And when you get new mutations, then it means that the genome sequence can change, and you can get these flips from a pathogen to a non-pathogen to a pathogen, or from not antibiotic resistant to resistant. So I'm just illustrating that here. These two people have the same species. Let's just imagine they have the exact same amount of this species of bacteria, but one of them has the green gene and the other one has the purple gene. If this is the difference between being a deadly pathogen and not, you would want to know not that you have this species, but that you have the purple gene versus the green gene. And in particular, these techniques from evolutionary biology and the study of how genome sequences change over time can help us get at this. Essentially, what we're looking for is examples where parts of the genome get cut out. So you lose functions by losing genes. You can get copies and pastes. So you can take a gene and you can make more copies of it. You can even copy from another species and insert it into this species. That happens quite often. And then you get typos where the sequence of the gene is still there, but its sequence has changed, and therefore it doesn't do what it used to do. So cuts, copy pastes, and typos are changing on a daily basis, even if our microbial ecology hasn't changed. How much does this actually happen? Are these copy paste errors and these deletion errors common? Here is some data we've looked at. We compared a bunch of different publicly available data sets from 8 different continents, 8 different countries across multiple continents. And for every pair of people, we said, if they have the same species, do they share exactly the same genes, or have some of the genes been cut or copied or pasted? And here's the result. These are a bunch of very common microbial species that you find across most people. And what you can see is that on average, and there's a lot of differences from one species to another, but on average, 25% of the genes are different from one person to another. So Deepak could have this species and I could have this species, but my version might have 25% of its genome encoding proteins that Deepak's species doesn't have, and vice versa. And if one gene can flip from a pathogen to a non-pathogen, you can imagine this amount of genetic difference is huge. And it's sort of amazing we even consider them to be the same species, because a human and a chimp are only 2% different. So does that matter? Maybe I flip 25% of the genes compared to Deepak, but it's just noise. It doesn't really make any difference. Let me tell you a quick story. This time not about my family, but about other patients. Imagine that you found out you had cancer. This is a huge and horrible piece of news to get. You go to your doctor and your doctor says, don't worry, there's an excellent chemotherapy drug that works almost all the time for people with this disease. So you start taking it, you go through a difficult therapy, you come out the other side, and you go in to get imaging, and it turns out your tumor is still there. It's grown, it hasn't shrunk. What might have just happened to you is that a microbe in your gut inactivated your chemotherapy drug. And this is a real story. There are a number of different drugs where this is happening, and it is really scary if we don't know about it. You could be getting the most precise, top of the line, best treatment, and it could just be getting turned off by your gut microbes. Similarly, they could be making a toxic side effect happen, or they could be making your drug even more active so that you're getting the wrong dose, a dose that's way higher than what your doctor thinks you should be getting. So with that example kind of in the back of our minds, I want to step back and tell you a summary of what we've been able to learn by applying these evolutionary approaches to a huge amount of data. And this is only possible because of our faster algorithms that we have. And this computation would still be running now. It would take years to run, even on expensive cloud computing. But we were able within a few weeks to analyze over 25,000 experiments that have been conducted by labs around the world, looking at people for diverse people from different populations, and have put their data in a public repository. We grabbed this data and we started looking for patterns. Here's a quick summary of some of the highlights of what we have found. So one interesting fact is that for some microbes, but not others, the DNA, the genes that are there and the typos in those genes of your microbial cells, not your human cells, can tell us where you're from. So this picture is showing places where sampling was done, and the color of the dots on it are the countries from which people come. And those dots are connected up by lines that represent the evolutionary relationships between the strains found in the different people. So two dots that are close in this picture mean that your microbe, your E. rectale, has a similar genome to that other person's. And what you see here is that there is a group of Chinese strains in yellow. There's two groups, two kinds or clades of this species that are found in North America and Europe, but they're mixed. Individuals from North America and Europe can have one or the other. And then individuals from South America and Africa have very different strains. And so for many species, you can really track even down to like specific islands in the Pacific Ocean where somebody is from based on the DNA of their microbes. So it has this forensic or ancestry information in it, just like our human DNA does. We can also use this detailed view of the microbiome to track transmission from moms to babies, from individuals within households, and between twins, which helps us disentangle the effect of the human DNA or the human biology from the microbiome biology. And transmission is a really hot topic right now, as everyone knows. We're in the middle of a pandemic. And so just the same way that DNA sequence mutations have been used to figure out where SARS-CoV-2 originated from and where the different transmissions have happened, we can do this with the gut microbes and microbes from other parts of the human body. We see that diet and urbanization are several of the biggest drivers of differences in what strains you have. And this is consistent with a hypothesis that's been around a long time called the hygiene hypothesis. What we see is that individuals living in industrialized countries and with higher hygiene, which of course has its benefits, we also see, however, a loss of good microbes. And this has been associated with inflammatory diseases, just like the ones that my family suffers from. We also can identify the specific strains and the specific proteins that are involved in transforming diet or drugs in the ways that I've discussed during the webinar today. And this is critical if we wanted to develop a test. We're all familiar with the PCR tests that are being used to detect coronavirus right now. We could make a PCR test to tell whether you would have a bad reaction, side effect, or an inactivation of a drug before you would take it based on your microbiome. And finally, the kind of maybe most exciting idea for me is that we could start to identify these pathobionts. And that what that word means is microbes that are found in lots of people and are normally okay to have, but in certain people under certain circumstances, I hypothesize because of specific genetic changes in their strain, those become pathogens. And I think this is where we really need to focus in the future to understand more fully how the microbiome is not just associated with our health, but maybe playing a causal role in it. If we can do that, if we can achieve this future vision of there being a direct, at least predictive, if not causal relationship between specific DNA sequences from microbes and our health, then we're in a position to start using the microbiome in precision medicine. And unlike using human DNA in that way, where it's just a diagnostic or a prediction, the fact that the microbiome is variable, changes from person to person, changes within a person from day to day, means that we could also take action in the other direction, that the microbiome itself could be manipulated for therapeutic purposes as soon as we understand these relationships. So with that, I'd like to conclude, thank my amazing team and our funding sources, and I'm looking forward to the discussion.
Thank you so much, Katie. That was wonderful. And just to remind everybody, feel free to go to the chat room and put any questions in there that you like, and I'm happy to field those. Katie, maybe to start off with, you mentioned an area that's, I think, of interest to a lot of people, which is how drug interactions occur with differences in people's microbiome. And you gave the example of chemotherapy, but so many drugs that we take by mouth that are more routine may be affected by our microbiome. Can you say a bit more about that? And what can we, even if we know that that's occurring, is there predictive value from the sequences that you can garner and that would tell us to treat somebody with more or less of a drug or a different drug? And how can we utilize that information?
Yeah, thanks. Great question. So there are several hundred, a little over 200 drugs that are known as of now to be modified in some way by the microbiome. That might, in a few cases, it means that they're actually not active. You take a pill orally and it doesn't do what it's supposed to do unless a particular microbial reaction happens. There are a number included in that 200 or so. There's some that get inactivated, and there are some that work therapeutically but cause side effects, in some cases very serious side effects. What the problem is, and what we're working on in my lab, is that even though we know the microbiome's involved, because you can take a stool sample and put the drug on it and it changes chemically, or you know that it gets changed in people's bodies, even though we know that, we don't know what microbe is doing it. We just know it's the microbiome, but we don't know what microbe or what gene is involved. And that's where these really precise sequencing-based evolutionary techniques come in to say, when I see a particular protein there from this species, then it happens, and when I don't, it doesn't. And once we have that, it will become predictive. There are a handful a few dozen cases where that has been established, and where it's established, it's highly predictive of what happens to the drug, both in the lab, but also in people. So I think this is a really exciting and promising area where I estimate that in the not too distant future, we would be able to do something as precise as predict the bioavailability, meaning I give you a particular dose, how much is actually going to be circulating in your blood. And I'm very confident we would be able to make predictions about whether you should or shouldn't take a particular drug.
Katie, when people take antibiotics, then the purpose of those is to get absorbed in our blood and kill any bacteria that might be in our bloodstream or in tissues. But in doing so, those antibiotics also can kill the bacteria that are in our gut, which make up our microbiome, and they changes our flora in our gut. Um, how when I know a lot of people think about that when they're taking antibiotics, and they try not to for those reasons because it does change it. What can people? How should they think about that aspect of changing their microbiome when they take antibiotics?
Yeah, there is a drastic and immediate change in your microbiome. Not all species, but many of them respond to antibiotics. Some of them go away and never come back. But the interesting thing is that as soon as the antibiotics stop, the vast majority of microbes that became undetectable during this course of antibiotics reappear afterwards. I do feel like that is a very intense and negative thing to take the antibiotics. But on the other hand, a lot of what was there does come back. What's been surprising, though, is that it's not always the same strain that comes back. So some species go away and then the same strain comes back afterwards. Other ones actually evolve during that process. Either the microbe itself undergoes a change, like acquiring an antibiotic resistance gene that would let it come back, but other changes too in its genome. And in some cases, it goes away. That strain probably got wiped out, but you just every day are taking in new microbes through your food that you eat and drink and things that you touch. As we all are very aware of hygiene right now in this global pandemic, just like a virus can get inside of you, new bacteria are coming in every day. When there's a healthy flora there, they kind of, in most cases, have trouble colonizing, but after antibiotics, these new ones coming in can colonize. So all that to say, I'm not a doctor, I'm not giving medical advice, but I would avoid antibiotics unless you really need them. When you need them, you should probably take them, and your microbiome will come back. It's probably a good idea to be cognizant of what you're recolonizing yourself with. Yeah.
And one of the questions from the audience was, does yogurt with live bacteria culture restore good bacteria?
Yeah, there's a lot that's been published on yogurt. It's a little bit controversial. It's certainly not bad to have yogurt, but it's not a panacea, I guess I would summarize the data that way. In general, I would just broaden it out from yogurt. I mean, just speaking generally, yogurt is an example of a probiotic, meaning something we put in our body that has live bacteria in it intentionally. We're doing it all the time unintentionally. In general, probiotics haven't had as much scrutiny as they need to be able to say for sure which ones are helpful and not. That is happening now. Many of them, the things in them, when a colonoscopy is done and we actually test if those microbes colonized a person, they didn't. So many of these probiotic techniques we would use don't have a long-lasting and permanent effect on our microbiome. But I still think there's some hope that recolonizing in a specific way could be a positive thing. I think we just need to learn how to deliver them and which ones to deliver a bit more precisely than the products that are available now. All that to say, if it doesn't make you feel ill or have any bad side effects, I don't think it hurts to do it.
Yogurt's not bad.
I like it.
I've got there's a technical question. What type of dimensionality reduction tools have you implemented to analyze large data sets?
Yeah, great question. So that actually is part of the key of how we got a task that cost thousands of dollars on a fancy cloud server to work on a laptop. So if I'm comparing specific species in my microbiome to Deepak's, and I want to know if it might function differently, like that mine might be pathogenic and his wouldn't, or mine would inactivate a drug and his wouldn't, I can ignore most of the genome, the parts that are the same between my microbe and Deepak's microbe. I only need to know the parts that are different. And so because we have now got a pretty good view of which parts of the genome differ more than other parts, we were able to create a new database where you're not searching the sequences along the entire genome, but just at the variable places. And for people who are familiar with this, this is on a computer, kind of the virtual version of a genotyping array. So anybody who's familiar with a very, workhorse of human genetics called the genotyping array, if we want to compare the human DNA of me and Deepak, we also don't need to generally look at the whole genome to get a pretty good view of it. We can look at very specific places that commonly vary between people. So we made on the computer the equivalent of a genotyping array, and just like a genotyping array is cheaper than doing a whole genome sequencing project, we can do a cheaper compute, basically.
Mm-hmm. Yeah, Katie, I know, but everybody may not know that you started your career early on in analyzing what makes us human, meaning what are some of the differences between our genome and the chimp genome. Is this approach similar to in any way to what you used, where you focused on what was different between those two species?
Exactly. So in 2006, when it became possible to do these sequencing experiments, the predominant models in the field and the ones that were popular and advocated by other people were these ecological models. And that would be equivalent to saying my microbiome has a chimp and yours doesn't. But it wouldn't say why, like what parts of the DNA are different. And so that's where these evolutionary techniques of what makes us human, why are we different from a chimp? Is it specific proteins we have that a chimp doesn't have? Is it mutations in those proteins? Is it mutations in the sequences of the genome that turn the genes on and off? Is it something else? Those kind of statistical models, probabilistic models that I helped pioneer for studying the human genome and what makes us different from a chimp, we can use those same evolutionary techniques to study the microbiome. And we were able to make a big impact in this field because this is a much less common approach to use than the ecological approach, but it's much more precise. And that's where I think it really holds the value for medicine. The ecology is very exciting to see species coming and going and sort of describe what's present in a microbiome, but it remains descriptive. It's not predictive, it's not causal, and it's not actionable in like a clinical sense.
Yeah, so that perfectly leads into the next question that we received, and that is what type of interventions could be used to change the gut microbiota for therapeutic purposes, and particularly around cross-communication between the microbiome and the central nervous system, immune system within the central nervous system or other aspects of the central nervous system.
Yeah, so just commenting on the central nervous system, there's a huge body of science now supporting the idea that microbes in our whole body, but specifically in our gut, are able to communicate with other parts of our body, including the brain. So how do they do this? They can do it using nerves. Our nervous system is connecting all of the organs in our body and connecting all of them to our brain. It's just like the flights around the world that are connecting all of us and leading to the spread of coronavirus. This is like a highway inside of our body connecting organs. So if you could send a chemical signal to nerves and nerves could communicate to the brain or to some other organ, that's would be and has been established as one mechanism. Another one is the microbiome produces a ridiculous amount of different chemicals, small molecules, and some of those leave the gut and circulate in our blood. So our blood is another highway around our body that can carry information. And then finally, some of the microbes in different parts of our body actually migrate from one place to another. An easy one to understand is something from your skin could end up in your gut because you put your fingers in your mouth. But a much more complicated one is that microbes in your gut could move to your pancreas and cause cancer. And so the microbes themselves can move around. So just kind of by background to answering the question, there's communication in a number of different ways. And then to the question of what they're communicating, I think that's pretty open still. There are associations with a number of neurological diseases, especially neurodevelopmental diseases. There, in almost every disease we look at, the microbiome is different. So in heart disease and neurodegenerative disease, it's different in people with different infections compared to others. But the big question is that cause or effect? So if I have an asthmatic body, does that give me an asthmatic microbiome, or did my microbiome actually communicate to my lungs in some way and cause asthma? And that, I think, is where we really need the hard science going forward to answer those causal questions. There are a lot of associations, and some, but not very many, have been shown to be causal so far.
And I guess the establishing cause and effect is the prerequisite or key to using this for therapeutic purposes.
Exactly. Yeah. I mean, it's important to understand, but it also is the prerequisite to taking action. Now, what kind of things could we do to treat was sort of the beginning part of that question. You could just start trying stuff. There's sort of two approaches to making a therapy. One is understand the problem and design something to fix it. I'm a big advocate for that. But the other one is trial and error. And this is how, a lot of therapies are discovered. Either trial and error in the lab or even trial error on willing people who want to try them. So a lot of people have taken probiotics after a course of antibiotics. You can collect data retrospectively and figure out, if that makes a difference in terms of some outcomes. So these population-based studies are also really, really important. I think they complement the more mechanistic ones. But there you're running the risk, you're taking the you're depending on chance to discover the therapy. You're going to try a lot of things and hope that you discover the one that works. But if you really understand the problem, the disease or the inactivation of the drug that you're trying to solve, you can engineer a solution much faster and probably a better one.
Mm-hmm. And in the absence of that kind of detailed information, are there things people can do to improve their gut health in general?
Yeah, it's a great question. Diet is the biggest driver of your gut microbiome. That is, it's a driver that changes on a pretty regular basis. So there's great data from my colleague Peter Turnbow at UCSF that if you take a heavy meat eater and you put them on a vegan diet, within 24 hours, there are drastic changes to their microbiome. And so, and then you go back to meat and a few days later, it switches back again. So you can manipulate the microbiome pretty effectively through diet, probably more so than in any other way. The question is, what is a healthy microbiome? There is some evidence that certain things in your diet are bad, and some of them we knew already. A really interesting story related to heart disease, which you might like, Deepak, since you work on the heart. We've all known for years that a lot of red meat is bad for your heart. It can cause atherosclerosis. But it turns out that the chemical process through which something in meat leads to risk is actually going through a particular bacterium that takes something called L-carnitine in the meat, transforms it into the chemical that chemical, not the original product in the meat, is what causes the increased risk. And that's been shown in mouse models and in human studies. And so I think that as we figure out more of these connections, we can learn not just which diets are healthy, but also why. It might be that they're healthy because they're promoting a certain kind of microbiome. So I hate to give health advice, but there's one: don't eat a lot of red meat. It's also good for the planet.
That's right. And just as there could be chemical things coming from the microbiome that affect the heart, in neurogenesis, we have a question: is it possible to figure out if the microbiome influences development of neurogenesis or creation of new neurons in the developing brain, or even in the adult, as there's ongoing creation of new neurons?
Yeah, I think there is evidence in mouse models and some emerging evidence in people that it can affect neurogenesis in adults or children. What is harder to figure out is what's happening in utero. There are not a lot of microbes. The placenta and has always been known to have some microbes, but it was considered that the baby was sterile. There's new data with more sensitive techniques showing that's not entirely the case. Not only is the baby not totally sterile, but there's also a lot of chemicals coming from the mom into the baby's environment that would change depending on the mom's microbiome. So this is an emerging area to try to understand how the mom's microbiome, the mom's diet, the mom's health might affect the developing embryo, not just the brain, but in general.
And there's another question here about phages. So is there knowledge about how phages, and maybe you could explain for everybody what that is, how those interact with the microbiome?
Yeah, so a phage is a virus. Coronavirus is a virus that infects people. Phages are viruses that infect bacteria. So just like we get viral infections, so do bacteria. And those viruses collectively are called phages. There is very good evidence that the phages in all kinds of environments, not just the human body, but the globe period, are big drivers of what bacteria you find there. Just like they can wipe out a human population, they could wipe out a bacterial population. So they're a very hot topic in microbiome research right now. They have been somewhat invisible in these data sets that I've been talking about for two reasons. One is they're very small, and if they're not actively replicating, they're not making a lot of DNA or RNA. They're not they're not often sequenced, even if they're there, or they don't make up very much of the sequencing that you do. So you have to really look for them. But amazingly, when people have looked for a few phages that are known, in some cases, it turns out they took up 40% of the sequencing file that came out of the computer. So sometimes there are a lot of them. And what would help us to find them would to have better descriptions of all the different phages that are out there. Just like I described that it's much easier to find a bacterium or a bacterial gene if we've seen it before in a bacterium that we've isolated and studied in the lab. If we had genomes and they were much better described for a lot of phages, then these same techniques I described today could be used for phages in principle. It's just that the data sets the databases and the sequencing itself hasn't been targeted to that problem yet, but it's happening right now.
Great, great. We're getting great questions here from the audience. It's clearly a very informed audience. Here's another one that we haven't talked about yet, but I know many people have heard about this, and that's around fecal transplants. So the notion of fecal transplants to transfer essentially the microbiome from one person to the other. The question is that still happening, and what are the data around that?
It is still happening, and I should have mentioned it as one of the ways you can change your microbiome along with diet and probiotics. We're involved as scientists, not as patients, in a couple of studies at UCSF that are looking at what happens in the context of fecal transplants. What I can tell you is that if you don't clear out the microbiome that was there before a fecal transplant, they don't have a big effect. They basically pass through you, and most of the microbes there don't colonize. And I guess I should have said, for folks that don't know, a fecal transplant is literally what it sounds like. You're taking poop from one individual and trying to put it in another person. And you can do that by a variety of modes of delivery. One of the studies we're involved in is looking at the best way to get it in there is with a colonoscopy. The prep itself and taking antibiotics, as I said, help that new microbiome to colonize the patient. And we're also looking at whether following up with either enema or some capsules, something that you could do at home after the colonoscopy, would increase the probability that the microbiome from the healthy donor would take over the gut of the patient. For Crohn's disease, this has been shown in some percentage of patients to be efficacious. We're now looking at several other diseases.
Mm-hmm. Great. Going back to phages for a moment, Larry, who you know, asked the question of, aren't phages able to be designed so they can target pathogenic viruses or bacteria?
Oh yeah, Larry, great point. This is an old idea that has just gotten more popular recently. More than 30 years ago, as people were starting to notice antibiotic resistance becoming a problem, there were a group of scientists who had the idea that instead of using drugs to kill bacteria, why don't we use phage? They're naturally designed to kill bacteria. It's called phage therapy. There were, as you can imagine, some concerns, both scientific and ethical, about putting a lot of viruses in people. There are a lot of naysayers, but it was never really proven to not work. And there are some case studies showing it did work. In this age with antibiotic resistance growing by the day and potentially becoming a huge life-threatening problem for many people, the idea of phage therapy is back in vogue, and there are some scientists who are looking at it. Like many things in science, it's sort of an outsider idea. It sometimes takes a little while to take hold, because it came up in the past and kind of got dismissed, but I actually think it's promising. So let's see what happens.
Great. Okay, let me wrap up with one more question, and that is, you alluded to earlier that everybody's microbiome is a little bit different, and some of that may be because there are alterations that go on in the species, but some of it is that intrinsically different, even at the very early stage. What are there data around whether it's an individual's genetics actually influence what the nature of their individualized microbiome baseline might be, and everybody's a little bit different in that respect?
Yeah, this has been looked at, and just like a lot of other traits, it's really helpful to look at twins or other related people to try to answer that question. So if you take identical twins and then fraternal or dizygotic twins, and then you could take siblings or other household members, people who are less and less related, ask if their microbiomes are less and less similar. And the initial observation was that they were. That as you got more genetically distant, you got more dissimilar microbiomes. But at that time, there weren't very large sample sizes for the identical versus non-identical twins. And it turns out a lot of that was just proximity, sharing microbes because you live in a household, and twins share a lot of things with each other, as I can say as a mom of twins. And so, including poop. So what's emerging now is that there are a few examples of specific microbes where your human genetic variation, your genetic makeup, and how that's different from other people plays a role in whether you have that microbe. But for many of them, it actually seems not to be very related to genetics. And that's in human studies, but also really importantly, animal studies, where you can control the food and the environment much better. There is a genetic component, but it's pretty small.
Great. Actually, we got one more question that came in, and I think we have time to maybe do this one as our last one. And that is around the microbiome and obesity and metabolic syndrome that often comes with that causes heart disease and diabetes and that whole complex syndrome. And what is the connection between those, and has there been anything demonstrated?
Yeah, this is one of the cases where some causality has been demonstrated. So people with metabolic syndrome, people who are obese, on average have microbiomes that look different from healthy people. They don't necessarily look different in a consistent way. There's sort of a range of variability in healthy people, and then people with these conditions are sort of outside of that range, but in different ways. Where causality has been established is through animal studies. Where you can create a mouse that's germ-free. It has no microbiome, and some, but not all of the microbes that live in people can live in mice if you start with a mouse with no microbes and put them in. So you can put a human microbiome in a mouse, and then you can evaluate things that are harder to control and evaluate in human studies. And what's been shown is that, amazingly, that you can take a stool from an obese individual and transfer some of those metabolic traits to a mouse, in terms of their blood work, weight gain, et cetera. And so in that case, it's causal. That mouse was not obese, didn't have a genetic predisposition to be obese, isn't eating more than other mice, but the trait gets transferred with the microbiome. That is a very compelling piece of evidence that the microbiome is causal. What isn't known is why. Which microbe is it? What are they doing? How are they doing that? And what's also true is that if you look in people, which are much more variable than laboratory mice are, you can't find anything in the microbiome that can really predict well that one person's obese and another person isn't. It's really hard to accurately predict their body mass index, for example, across hundreds of people at a population level. So there is some causality, but there's also a lot of complexity. And that's, I think, where these precision techniques are going to come in, to really pinpointing the why, and then we'll understand it and we can hopefully correct it.
Great. Well, thank you so much, Katie, for a very fascinating discussion. And thank you to the audience for your questions and your attention. I hope you all enjoyed our discussion today, and look for our next Newton series talks. Please feel free to communicate with us if you want to go deeper into any of these areas from today's talk or previous talks, and we would welcome an interaction with you. Our Gladstone URL is posted in the chat room. We invite you to come visit our website and contact us if there's anything you want to learn more about. We're here to engage with you. Thank you all very much, and please join me in thanking Katie for a wonderful discussion.